Welcome back to Immunology, and today's submodule is vaccines. A subunit of vaccine is that shows, so you have a virus that contains DNA, RNA, or bacteria that contains a specific part or fragment of the membrane. You can genetically engineer the antigen for that particular piece of protein or that membrane into a capsid. You place that gene into a plasmid, and you let a foreign microbe like a bacteria engulf it, and you express it. You could then purify that protein or purify that toxoid. These antigens are able to stimulate immunity without the whole pathogen being present. Attenuated vaccines are as follows. Viruses that are grown in a lab in a cell type which is not usual to its normal host. So for example, if you were to take the polio virus that normally is going to be infecting human nerve cells, and you put that into a monkey's kidney cells. When the virus is grown in a lab, it accumulates mutations which could potentially change it and even weaken it. So as a result, you do cripple the virus so that it's not as dangerous, but you may also run the risk of infecting other kind of cells, such as infecting antigen-presenting cells. Vaccines for measles, rubella, mumps, MMR is created as so. They generally will create a pretty long-lasting immunity, and they can produce and induce memory killer T cell activity. However, the drawbacks are as follows. They're extremely risky for those that have a weakened immune system. And in case the mutation turns in the benefit of the virus, it could restore the strength of the virus and make it more dangerous. You could take the killed or inactivated virus via heat or chemicals when it's dead, but the antigenicity is still present, and administer that. You could also have a live attenuated cell or virus on the bottom where the virulence has been reduced or eliminated and is still alive. It still retains the same antigenicity, and then the vaccine microbes can multiply and boost the immune system that way. A carrier vaccine is one in which the vaccine preparation utilizes genetic engineering to introduce a gene or genes pathogenic microbe into a virus that doesn't cause disease, usually the DNA RNA vaccines. Think of this as a Trojan horse concept, where it carries a gene of a pathogenic microbe into the human cell. The carrier vaccine should not generate any memory killer T cells, and there's also no chance that this vaccine will cause disease because it doesn't have all the genetic information to code for it. Only one or few of the pathogen's many genes is carried in this vaccine. So the drawbacks to this is that there is quite a bit of theory. It's very costly to design, and this is the foundation of the SARS-CoV-2 vaccine. So DNA that codes for protein antigen is extracted from the pathogen genome. That will then be cloned into a plasmid vector, amplified, prepared into a vaccine, injected into vaccine, and hopefully the cells are going to recognize it, read that fragment of DNA, express that protein, and now you have the ability to stimulate an immune response. Vaccines need to mimic the invasion of a pathogenic microbe, and the whole point is for the immune system to view the vaccine as both foreign and dangerous. It's not a problem when a vaccine is a crippled virus because it's still a virus, but vaccines are composed of only one microbial proteins will need an adjuvant. Adjuvant is oftentimes aluminum hydroxide or alum that is added in as an additional ingredient to help the immune system recognize it as foreign. For example, in the Cervix vaccine for the human papillomavirus, it uses MPL, monophospholipid A, which is a common ingredient added as an adjuvant. The viral coat proteins provide the first signal, and then the MPL alerts the immune system as another foreign entity in the body, so it alerts the immune system to be a little bit more aggressive. Here are some key terminologies in this module. The conjugate vaccine is going to be a vaccine composed of a weak antigen coupled with a stronger antigen that can activate a protective primary immune response. Inactivated vaccine or vaccine composed of chemically inactivated, irradiated, or heat-killed pathogens. Live attenuated vaccines are those that compose of pathogens that have not been inactivated but is incapable of normal survival in the host. Herd immunity is roughly about 83 to 94% of vaccination level, which will then prevent the spread of infection within a population. Subunit vaccine is when a vaccine is composed of a cell surface antigen of pathogen that's designed to stimulate the production of immunoglobulins that block pathogens' interaction with target cells. A toxoid vaccine is a vaccine that utilizes an inactivated endotoxin, exotoxin, that is employed to stimulate the adaptive immune system. Adjuvant is a component of vaccine that's used to enhance the antigen-specific immune responses. helping you to boost that inflammatory response required to have a complete adaptive immune system. All right, thanks.